Hello, Yulan, and welcome to your first revision video from science. And what we are going to be doing in these revision videos is giving you a little bit of an overview of some of the key aspects of each of the topics in your course and looking at those through the lens of some past exam questions that will allow you just to try and recall some of the key information that's required to answer questions, but also look at how we interpret different questions in each of the topics across the course and how we deconstruct and decode them to make sure that actually you're able to feel as fully prepared as you possibly can for your exams. So, as this video is titled, Cell Biology, this is our first topic from the GCSE course, and obviously our first video we're going to go through. In terms of what you need, you'll see that in terms of what I've got here, I've just got pen and paper. You'll have access to these past paper questions, and feel free to work through them alongside me as we go through this tutorial. And at the end of this tutorial as well, you'll also see and be provided with some access to some links to some other practice questions in and around this topic area and answers that you'll then be able to do under your own steam. So I'll dive straight in. And as you'll see, we are starting off with a piece of familiar kind of equipment that we know relative decent amount. And that is, as you can see from our first question, a microscope. So as you can see, our question says a student prepared some animal cells to view using a microscope. Figure 1 shows the student preparing the cells. Now, the question, as you can see, I've highlighted an important bit. Name 2 pieces of laboratory equipment, alpha highlighter, that the student could have used to prepare cells to view using a microscope. Now, in terms of what this question is asking you, and the reason why I've highlighted these 2 things is because Figure 1's there for a reason. It's not just there to look pretty, not just there to look nice. What it is, it's there to give you kind of a little bit of a clue. In that it gives you 2 pieces of equipment that you already know the names of, that you know are needed to prepare cells so that you can see them under a microscope. And as you can see, piece 1, piece 2, 2 easy answers, is a slide, and also a cover slip. But if you think back to the practicals that you've done in past in using microscopes and other different things that might not be on Figure 1, but would also be legitimate, think about the use of iodine to observe cells, to make them clearer. We use dyes, we use stains. So, for example, iodine is a really good shout. In terms of putting those dyes and stains on, we need to use pipettes, don't we? To drop that liquid on over our sample to make it a bit easier. When we've done onion cells, we also need to remove the cells, remove thin layers of cells. If you think back to your very first required practical, you might use tweezers for that. And also to cut up samples so that they become quite small. What piece of equipment do you know that we use to cut things up, to make things small in size? Use scalpels. So immediately you've got a variety of different bits of equipment that you can use straight away, straight off the bat, and it gives you a really good in to this question. So we'll continue on. Next question below. Figure 2 shows the student's light microscope. Now, if you just have a little look at this question, pretty standard. The only difference compared to the light microscopes you use is that here we've got a mirror. Doesn't affect process in any way. Instead of generating the light itself, it reflects light up in through the sample that's on the stage, just like it does in the microscopes that we've used in class. So another couple of key questions here that are relatively simple to deal with. First one, name part A. What we've got to do is try and remember the parts of the microscope. Part A obviously is here. As we know, we put our eyes on this part, having done this before. We know that there's a piece of glass here that we look through. We call those pieces of glass lenses. So you put 2 and 2 together. Hopefully, if you've put 2 and 2 together, you should be able to recall that this is the eyepiece lens. Now, part B doesn't ask us for the name, but if we just recall that the name of this, in terms of its function, is the coarse or fine focus wheel, What's its function? Now, remember we've spoken about function. There's another word being job. So what's its job? What's the job of part B? Now, what we don't want to be talking about is we don't want to be talking about magnification, because that's not correct. That would be if part B was labeled here, which is our objective lenses, which we know we change in order to alter the magnification. So that's not what we're looking for. What we're looking for is, as you know, when you move the fine and coarse focus wheel, it moves the stage up and down. And from your experience with microscopes, what does that do? Well, hopefully you should recall that actually it helps to focus the image. Now, we might have different ways of saying that. Could say it makes it clearer. We could say it improves the resolution. All of those are absolutely fine, saying the same thing. But we might also just go for a quite literal answer in that we've kind of given it already. What does it do? We turn that, it moves this up and down. Another answer for this is that it moves stage up and down. Great. So we keep going. And hopefully that sort of starts to give you a little bit of a starting feel for practicing these questions and making sure we're recalling this information. So you have a look at our next question. Just readjust the visualizable. The student looked at the cells using the microscope. Suggest one reason why the student could not see any cells when looking through part A. So it doesn't give us anything obvious in the example. So there's lots of different ideas that we could include here. And this is thinking about when you've done microscopes and used them, what's gone wrong? Well, slide might not be in the correct position. The objective lenses that we mentioned might not be in place over the slide. And then we could also start to think about other reasons that the objective lens might be dirty, for example. Another reason, I can't see it, might not be in focus. So microscope might not be in focus. A whole variety of different reasons as to why our image might not be able to be seen. So continue on. Now, question changed tact slightly here, whereby it starts to talk about specialized animal cells. It gives us red blood cells. Now, remember, specialized cells, they have unique features carry out unique or specific or specialized jobs. So we've got a command word, and we've talked a lot about command words in the last few months. Compare. What does compare ask us to do? Well, it asks us to compare similarities and differences. So we need to make sure we're including both because we've got that command word. And our two cells it's asking us to look at are red blood cells and plant cells. So what do we know about the structure? What do we know about the structure of each of these? What's similar and what's different? Now, it's a 6-mark question. So we've got to think about how we're going to try and break down the marks. And as we've talked about before many times, if it's a compare question and it's a direct comparison, let's make it easy for the examiner to see what it is that we're trying to get across. Similarities, differences. And we can put it directly in a table. So if we put it directly in a table now, so similarities and what are our differences? Now, let's start with similarities. Now, we don't necessarily have to have the same number of both because it doesn't ask us to have that, but we do need to make sure we have a bit of both. Now, think about features that they both have. Now, I guess in some respects, it might actually be easier to start to think about differences because we've talked a lot before about one of the most unique features about a red blood cell. And the red blood cell, its most unique feature, is that it has no nucleus. But the plant cell does. Now, what do we know about plant cells versus animal cells? We know features that they have that animal cells don't. We could probably come up with another one quite quickly. And if we think about outside of plant cells, plant cell has a cell wall. Red blood cell doesn't. Okay, good. Starting point. No nucleus, cell wall. Do we know any other cell structures? Cytoplasm, cell membrane. Is there a similarity there that we could pull out? Well, yes, there is, because both have cytoplasms. Both have cell membranes. So we've talked about the structure and got 4 points already. Then in some respects, it's probably easier to go back to differences because they stick out more prominently. So let's see if we can come up with 2 more differences. Now, you might remember from doing the red blood cell, the teacher or your textbook, your revision guide that you've looked at, let's talk about the shape of a red blood cell. And if you remember the name of that shape is that a red blood cell is a biconcave disc. Whereas a plant cell isn't. It's another difference. It doesn't need to tell us why one's biconcave disc, why one isn't. It's just asking us to say what's the similarity and difference, not why it's one particular feature or one particular feature. And then go back to the other side of the similarity and difference. Go back to plant cells. Go back to that idea of what we know that plant cells have that animal cells don't. Plant cells have chloroplasts due to photosynthesis. But red blood cells don't. Now, there's a whole other multitude of reasons that you could put here. These aren't the only 6. You might talk about the fact that red blood cells contain hemoglobin. Plant cells don't. Red blood cells don't contain chlorophyll. Plant cells do contain chlorophyll. We can talk about that difference between chlorophyll and chloroplasts. Red blood cells have no permanent vacuole, whereas plant cells do have a permanent vacuole. And then size, red blood cells are much smaller than plant cells. So there's lots of different possible answers there, but this is a really clear way to present it so an examiner can see exactly what you mean and exactly what you mean. So let's continue on a little bit down here. When placed into a beaker, a red blood cell bursts. A plant cell does not burst. Explain why the red blood cell bursts, but the plant blood cell does not. Plant blood cell. What am I talking about? The plant cell burst. Now remember, command word, explain. Hopefully you're recalling. Whenever you see explain, we need to be thinking about what, but also why. Give a reason for what's happening. So why does this burst and why does this not? This is what the question is asking us. Now, water, cell biology. This should be ringing a bell. We should start to be thinking about how does water move? Because when placed in a beaker of water, a red blood cell bursts. Something bursts, there's too much inside of it. So water must have moved in. Whereas this hasn't burst. So there's been no water moving in. So what do we know about water moving? Well, think back, look through your revision notes, and you should be able to remember that water would enter the cells by osmosis. Water would enter the cells by osmosis. Now, this would fill the red blood cell and cause it to burst. So it's swell, burst. But the plant cell doesn't. So what's the difference between the plant cell and the red blood cell? That lets things come in and out of its structure. Well, they both have cell membranes, talking about that earlier at the top. But what do they not both have? Well, they don't both have a cell wall. Only a plant cell does. So the plant cell has a cell wall. And what does that do? It's going to stop the water moving in by osmosis. And stop the water moving in by osmosis. Good. Let's continue on. Just looking at these ideas in a little bit more detail now. So we've got this idea at the top. Question starts with question 2. Now, very first part of the question here. It says diffusion is an important process in animals and plants. What's meant by diffusion? Well, it's just another way of asking for a definition, isn't it? What's meant by diffusion? So think back and try and recall that definition. We've got 2 marks. So we know that it's the movement, or we could say it's the spreading out of particles. You might say things like molecules, substances, chemicals, gases, liquids. Doesn't overly matter. Quite a few different things. But here's the key thing for the second part. From where to where? So it's that movement of particles or spreading out of particles from an area of high concentration. There's lots of those particles to an area of low concentration. Now, sometimes that can be quite a wordy sentence, and an easier way to try and remember that, or shorten the way we say that, is they travel down a concentration gradient. That means the exact same thing. Good. So let's move on. Figure 1, which is part of the leaf. So hopefully you can start to remember that these are different specialized parts of the leaf. Molecules of carbon dioxide diffuse from the air into the mesophyll. So that carbon dioxide, as it says, diffuses from the air into the mesophyll here. It's talking about that. Which 2 changes will increase the rate at which carbon dioxide diffuses into the mesophyll cells? Now, the reason why I've highlighted 2 is just when it comes to exam questions, this is easy marks, guys. Don't just tick 1, even if it's a tick box, even if you don't know. Just filling it out and visualize them. Even if you don't know. Always look at that number, take the one you know. So you can't, if you don't know another, just take another one anyway. So what are options? To increase the rate at which carbon dioxide diffuses. So what are things that increase the rate, factors that affect diffusion? That's what this question's asking us. One of those factors that affects diffusion that you'll find in your revision guide is an increased concentration gradient, which points us to this one. Because if we've got more carbon dioxide on the outside, then what we end up with, less carbon dioxide inside, we end up with a greater concentration gradient. Which means we've got a faster rate of diffusion. Now, the other thing is obviously that carbon dioxide's got to come from somewhere. Part of it's coming from the air, from the stomata, through the holes. Now, if you want more carbon dioxide moving into the cells, you've got to get it from somewhere. You've got to get it from the air. So it's going to be the stomata as well. More open stomata going to allow more CO2 to diffuse in. Good. Then move on to the last couple of questions here. Down the bottom, you see it says diffusion also happens in human lungs. Figure 2 shows the human breathing system. Explain how the lungs are adapted. Explain how the lungs are adapted for efficient exchange of gases by diffusion. So, remember that, explain. What and why. So in this case, what it's asking us is, what are the features of the lungs that allow for efficient gas exchange, and why or how do they do this? Now remember, we said figures are here to help us, to give us ideas to jog our memories. Specifically, this. Got loads of marks right here on the page. We've just got to try and remember them and try and call them. So if we think about the structure of the lungs and we think about these tiny little air sacs that come at the very bottom, right there, we can get a mark. We've got many, what are the names of them? Alveoli, not ravioli. That's a feature, but we haven't said why or how it helps for efficient gas exchange. Well, if there are many of them, it means there's a greater surface area. And if there's a greater surface area, obviously means that there can be more gas exchange. Greater surface area of the lungs. Now, even gives you a word here, capillary. Capillaries, as we know, carry blood. That gas, that oxygen has got to get into that blood, and that carbon dioxide that's in that blood has got to get out into the lungs. So what do we know about capillaries? Well, they're thin. So that's a feature, but why is that a useful feature? Sometimes they might say they're 1 cell thick. Why is that a useful feature? Well, it means there's a short diffusion distance for carbon dioxide and oxygen. What else can we think of? Because right now we're doing really well in that we've got relevant points that are identified and we're linking them to explanations. So we're explaining what's going on. So we could come up with a couple more. Now, we need lots of blood. We need it quickly. So this idea that there are lots of capillaries or what we might say is that there's a good network means that we've got a good blood supply. Now, why is that useful? Because you can bring in and remove CO2 and oxygen quickly. Not just the amount of it, but it's the speed at which it can go as well. And then finally, what else is useful? Well, it's this idea that it's this bit about it's our ventilation mechanisms. It's the fact that oxygen, air in oxygen, carbon dioxide air can be moved in and out of the lungs quickly. So breathing keeps the lungs ventilated. And if it keeps the lungs ventilated, it means you can remove CO2 from the body and bring in oxygen quickly. Loads of possible marks there. Hopefully gives you a bit of an overview and explanation around the lungs. And then the final question. So down the bottom, figure 3 shows a root hair cell. So we've got a diagram. Figures are important. Remember that. Name the process by which water molecules enter. Water moving, enter, 1 mark. Has to be osmosis. Has to be osmosis. But the second question is a little different. Nitrate ions need a different method of transport. So, not osmosis, this question. The answer to this question. Not osmosis. We only know 3. Diffusion, osmosis, but hopefully remember active transport. So not that, this question. Explain how the nitrate ions in figure 3 are transported into the root hair cells. So you see I've highlighted them in orange just to make them a little bit more obvious. Again, use figure 3, it even tells you your answers here. So you're looking for your answer. It's one of these 2. And this gives you a clue. Now, hopefully you should notice that if I highlight a few more of these, or X's are nitrate ions, where is the concentration of these nitrate ions largest? It's already inside. But the question's telling us that these nitrate ions move into this root hair cell. So it's moving from a low to high concentration. What does that tell us about which one of these we should be talking about? Well, we already talked about diffusion, osmosis, high to low. So it's not diffusion. Process is active transport. We've got a mark. So what's our explanation? Well, it's low to high concentration. So there are a lower concentration of nitrate ions outside the cell. They need to be moved to inside where there is a higher concentration. Great, lower concentration to a higher concentration. But remember, active transport, what does it need to do that? To do this, remember that word active implies something. Passive also implies something when we're talking about cell movement. To do this, it needs energy. So here, 1 mark. Lower to higher, 2nd mark. Energy, 3rd mark. So you've got a little bit of an overview there then of these different questions focusing on cell biology should help you to recall some of the key content around cell biology. Use print out this and work through it yourself. Give yourself some revision notes from it, and then use the links provided along with this video to have a go at some of the cell biology practice questions yourself, access the answers, and then you can speak to your teachers individually, drop them messages on Satchel One, drop them emails, come and speak to them in class, just with any areas which you struggled with attempting these questions, with the explanation of these questions. But hopefully these videos will be really useful to you. Look forward to doing more of them for you as we go through this year. So take care, folks.